students. So this is your midterm 2 study guide for microbiology. The bacterial colony. When bacteria cells are grown in a petri dish like the one shown here, they are grown on agar. One single bacteria cell will reproduce using binary fission, and they will give rise to an entire colony. A colony are made up of genetically identical bacteria cells that are all generated from a single parent cell. Bacteria have specific requirements for growth like all living things. The process of metabolism requires these 3 things: a source of carbon, a source of energy, and a source of electrons or hydrogen atoms. There are 4 classes of organisms based on what they use as a source of carbon and what they can metabolize for energy. We've got photoautotrophs, chemoautotrophs, photoheterotrophs, and chemoheterotrophs. When we're looking at these terms, it's best to look at the classifications by breaking down the words individually. For example, each of these terms begins with either photo or chemo. So here is the rule. The first word is going to define that organism's energy source. The word photo means light, as in sunlight. This means that organisms classified as photoautotrophs or photoheterotrophs are going to use sunlight or photons, which are packets of light energy, as their energy source. The word chemo means chemical, but for our purposes, I want you to think of chemo more specifically as organic chemical compounds. When you see the prefix chemo on these terms, I want you to know that these are your chemoautotrophs and your chemoheterotrophs. These are going to use organic chemical compounds as their energy source. These would be your proteins, lipids, and carbohydrates. Next, let's look at the second word that we see in these classifications. We see that each of these terms either has auto or hetero in the middle of it. So here's the rule again. The first word is going to define the energy source for that class of organisms. The second word is going to define the carbon source for that class of organisms. The word auto means self. These are your autotrophs. We know that these make their own food. They do not need to consume organic matter like heterotrophs do. So organisms that are classified as photoautotrophs and chemoautotrophs will use inorganic chemical compounds like carbon dioxide as their carbon source. The word hetero means other. These are your heterotrophs, which means eat other, but you can think of it as consuming organic matter in order to live. So this means that your photoheterotrophs and your chemoheterotrophs must consume organic molecules in order to use them for their carbon source. And remember, when I talk about organic molecules, we're referring to proteins, lipids, and carbohydrates. When we're talking about bacteria growth, it's important to look at some of the trends of the growth curve that we see here. When we analyze bacteria growth, it's really convenient to analyze it according to graphing the number of bacteria versus time. And when you do this, you create what's called a growth curve, like we see here. The growth curve has 4 different distinct phases: the lag phase, the log phase or exponential phase, the stationary phase, and finally, the death phase. In the lag phase of the growth curve is generated because the bacteria need a little bit of time to adjust to their new environment. The cells will not undergo binary fission until they've sufficiently adapted to their new habitat. In the log or exponential phase of the growth curve, here is where we see the maximal amount of growth for that bacteria. We also call this the active growth phase. The stationary phase of the growth curve is going to be a time in which the exponential phase or log phase basically levels off. So what's happening here is we're beginning to see evidence that space is running out, nutrients might be running out. Also, toxins can be building up in the culture that we're housing these organisms in. So what will happen is we'll end up, the log phase or exponential phase will give way to what we call the stationary phase. In the stationary phase, the bacteria cell number remains relatively constant. This means as many cells are dying as many cells are as being created. So the population stays fairly constant during this brief period of time. We do see, though, that this doesn't last forever because given enough time, you will definitely see a depletion of nutrition, a depletion of space, an extreme buildup of toxins, and you will no longer be able to have life in that culture. And at that time, you'll end up with more cells dying than being produced, and you will end up with this death phase. And the death phase will continue until all the bacteria have died. Okay, so next let's get into our genetics. So there are 2 functions of DNA, regardless of the organism in which it is found. DNA has an autocatalytic function, which means that it directs its own synthesis, and it also has a heterocatalytic function, which means it directs the synthesis of RNA and proteins. Let's take a moment to review the structure of DNA before we continue. So DNA is composed of a string of nucleic acids. An individual nucleic acid, like shown here, is composed of a phosphate group, a ribose sugar, and a nitrogenous base. The nitrogenous bases that are found in DNA include just these 4: adenine, guanine, cytosine, and thymine. When we discuss nucleotides, nucleotides come in 2 different forms. We have DNA nucleotides, and we also have RNA nucleotides. So nucleic acids are the building blocks for both of our DNA and RNA molecules in the body. And not just ours, but bacteria too. So RNA and DNA nucleotides both contain a phosphate group, and we see the phosphate groups here, and those are the same in both of those. Now, RNA and DNA nucleotides both contain a sugar molecule as well. However, in DNA, we see that the sugar is a deoxyribose sugar molecule. So at the 2 position, I do not have a hydroxide. Okay. Whereas in the RNA nucleotide, I have a ribose sugar. So I do have a hydroxyl group here. So if hopefully you've already made the connection, but in case you haven't, so this is why DNA is called deoxyribonucleic acid. It's because of the deoxyribose sugar that makes up the nucleotide. And RNA stands for ribonucleic acid because it has the ribose sugar. So nucleic acids are the building blocks for both DNA and RNA in all living organisms. In DNA, like I mentioned previously, you have the 4 nitrogenous bases, which are adenine, cytosine, guanine, and thymine. In RNA, we have one difference. In RNA, we're still going to have adenine, cytosine, and guanine, but instead of thymine, RNA does not have thymine. Instead, it will substitute that with a nitrogenous base called uracil. The backbone of DNA is actually made of alternating sugar and phosphate molecules, as shown here, that are strung together. DNA is double-stranded no matter what organism it is found in. So this includes our bacteria. Each of the strands of DNA are connected to each other through hydrogen bonding. And the hydrogen bonding occurs here between the nitrogenous bases that are facing each other towards the interface there. These bases are not allowed to just pair any way that they want. There's actually a rule. We only can have A and T, which is adenine and thymine, paired up together, and they will create 2 hydrogen bonds between them. And C and G are allowed to pair up as well, and they have the structure where they will create 3 hydrogen bonds between their structures. So why hydrogen bonding? Hydrogen bonding, which hopefully you remember from chemistry or from your introductory biology classes, hydrogen bonding is extremely weak. So why would we want such a weak bond for such an important molecule? Well, it's simply because we want this to be temporary. It's important for us to be able to break those hydrogen bonds whenever the cell or the mRNA or whatever machinery needs access to the DNA. It needs to be able to get to it when it needs it. So, for example, the 2 DNA strands have to be separated whenever you're going to have DNA replication during cellular reproduction. Also, anytime you're going to express any genes in the form of protein, you first have to do transcription, which means you're going to take a template of the DNA molecule, and you need access to that at that time. So that's why it's hydrogen bonding, so that we can easily have access to each one of those individual DNA strands. So the central dogma of molecular biology simply states that how our genetics, how our DNA does its job in the body is by expressing itself through transcription, followed by translation, and it ends up in a protein form. So it begins as DNA sequences that we call genes, and these genes are going to code for specific qualities or traits. The DNA will then be transcribed into RNA, and then it will go to a ribosome where it will be read as a triplet code, which is called a codon, and it will get translated into individual amino acids. Those amino acids are going to get strung together and form a protein, which will have a function in the body. Also, another important thing, the double-stranded DNA is also anti-parallel. So what that means is if you look at the sugar molecules, you'll see that there's one side of the molecule is 3' and the other one is 5'. So that's where we get the designation of when we say that it's oriented from 3' to 5' versus 5' to 3'. That's where those numbers come from, in case you're interested. But the DNA strands are oriented what we call anti-parallel to one another. So one end of the DNA molecule is going to be designated as 3', the other one as 5'. So one of the strands of the DNA molecule are going to be oriented from the 3' to 5', and the other one would be from 5' to 3'. You'll see why that is important here very shortly. We'll discuss that. So real briefly, let's just remind ourselves what happens in eukaryotes, just for contrast. So in a eukaryotic cell, like our cells, which are animal cells, the genetic material is in the nucleus of the cell. The DNA holds the instructions for all of the proteins that are required for all the functions. The instructions are coded as sequences of base pairs or nucleotide sequences that are on the individual nucleic acids that make up the DNA of the cell. Okay. Now, in prokaryotes, are bacteria are prokaryotic. Okay, they are much smaller than eukaryotic cells. Prokaryotic cells do not have a membrane, and they do not have any membrane-bound organelles. No nucleus, so the DNA can't be found in the nucleus. So where we do find the DNA, though, in bacteria cells is we find it in a region that we call the nucleoid. Now, everything that's alive does reproduce, and bacteria also reproduce. They reproduce using binary fission, which is an asexual process. It does not have any. It does not induce any genetic variability whatsoever, but nonetheless, they do have to reproduce. Otherwise, we wouldn't get colonies, and they would have died out a long time ago. So in order for this cellular cellular reproduction to occur, we first have to make a copy of the DNA. So we call this DNA replication. Sometimes this is also called DNA synthesis. So let's look at some of the compare and contrast some of the differences in the genome here. So the sizes of the genome are considerably different. They're about 50 times different. So the eukaryotes are going to have a genome that's about 50 times larger than bacteria, and the bacteria are going to have a genome that's 50 times smaller than eukaryotic cells. The DNA in bacteria cells consists of one single circular chromosome, which lies in the nucleoid region. Whereas in eukaryotes, eukaryotic DNA is linear. It's not circular, and it has more than one chromosome, so multiple chromosomes. So the location of the DNA, it's in the nucleoid region, which is in the cytoplasm. So we could say nucleoid or cytoplasm, but nucleoid is a little bit more correct here. Whereas DNA replication in eukaryotes is going to occur strictly in the nucleus. So DNA replication is a really fast process, relatively speaking, um, in bacteria cells versus what we see with eukaryotes. Eukaryotes, it's a more time-consuming process and more complex as well. So in eukaryotic cells, this is actually done during the S phase or synthesis phase of the cell cycle. So this occurs before mitosis begins. Okay, so let's look at what we call the origin of replication, or the ORIs. So when we look at DNA replication in bacteria cells, we see that the ORI is at one single point, and then it is going to go out into two different directions, and then it will continue around that circle until replication is complete. Now, the DNA in eukaryotes is much, the genome is huge by comparison. So we would never just be able to have this done by one single ORI. So in eukaryotic cells, we actually see up to even thousands of different points of origin, or ORIs within the entire genome. So big difference there. Okay, so some of this I've already said. So the bacterial genome consists of a small single circular chromosome, whereas eukaryotes are going to have large linear chromosomes and multiple chromosomes. The replication of prokaryotic DNA is much more rapid and can be up to 1,000 nucleotides per second, which is pretty impressive. The replication of DNA is much slower in your eukaryotic cells. Reproduction in eukaryotic cells is whenever you have a organism, eukaryotic organism, well, whenever you have an animal that is reproducing, that animal will reproduce using sexual means in which you have genetic recombination, like in us, for example. For that reason, animal cells are diploid, meaning that we have one full set of chromosomes from each parent. So two sets total, that's where we get the di from, di meaning two. Whereas bacteria do not have two parents, they only have one. Bacteria are haploid. They reproduce by a process called binary fission, which induces no genetic recombination, so they are just producing clones. Some species of bacteria have an additional small extra chromosomal circular DNA loop in their cytoplasm called a plasmid. Interestingly, these plasmids do not carry any of the genes that are absolutely necessary for the normal processes of life, but we do see that these plasmids carry genes that might alter the sensitivities or the abilities of the bacteria that have them. So these, the genes that are on plasmids are considered accessory genes because they are non-essential. So some of the abilities and traits that we see carried on these plasmids would be antibiotic resistance, habitat tolerance, and we see all sorts of different tolerances out there. The more we look, the more we find. And then bacterial conjugation, which we'll talk about a little bit later. And bacterial conjugation allows the exchange of genetic material between bacterial cells. So of genetic material between bacterial cells. Okay. So in our bacteria, DNA polymerase 3, or sometimes just abbreviated here as DNA Pol 3, is the enzyme required for DNA synthesis or DNA replication in our bacteria cells. And what DNA polymerase 3 does is it's going to add individual nucleotides according to their complementary bases. So remember that A is going to pair with T and C is going to pair with G. Okay, a word of caution before we go ahead. We're going to use some of the same terminology for different processes. So I'm going to go briefly, very briefly, over DNA replication, transcription, and translation. All three of these different processes have the same names for the same 3 steps. So each of these has an initiation step, each of these has an elongation step, each of these has a termination step. So I'll try to make sure that I keep that straight for you as well, but I also wanted you to be aware of that so you can keep track of that and hopefully not get confused by the repetition of terminology. Okay, so first let's talk about DNA replication and the initiation step. So how does this all begin? So initiation in bacteria cells is going to begin at the origin of replication. The origin of replication is a very specific nucleotide sequence. There is an enzyme called topoisomerase 2, sometimes called gyrase. And what that does is that's going to uncoil the DNA so that the other factors can have access to it to do their job. We also have helicase. Helicase is another enzyme that is going to function to separate the DNA strands by breaking those hydrogen bonds that we were talking about earlier. So we also are going to get 2 replication forks. We only are showing 1 here in this picture, but you'll get 2 replication forks, 1 in either direction from that replication bubble that occurs right at that point of that origin of replication, or ORI. The DNA near each replication fork is going to be coated with single-stranded binding proteins. There actually are some here. They're not labeled, but those are those small pink things here. And what that does is those single-stranded DNA proteins are going to inhibit that the double helix from rewinding again, because it's going to want to supercoil up on itself. So it prevents that from happening. Okay. So next we have the elongation step in DNA replication. DNA polymerase 3 can only synthesize DNA in one direction, and it goes from the 5' to 3' direction. But like I mentioned very briefly earlier, DNA is double-stranded, which means one strand is going to be oriented in the 5' to 3' direction, but the other one will be switched, so it would be from the 3' to 5'. So the consequence of this anti-parallel configuration, coupled with the factor that DNA polymerase 3 can only synthesize DNA in the 5' to 3' direction, is that one of those DNA strands are going to be replicated continuously, but the other one will have to be replicated discontinuously. So what we get here is we get what's called a leading strand, which would be your strand that will be continuously replicated. And you'll have a lagging strand, and the lagging strand will be discontinuous. So what will happen is you'll get small segments at a time that are replicated. These are called your Okazaki fragments, and then later on those fragments will be put together. Next, let's look at the steps of elongation in our DNA replication. So DNA helicase is going to open up the replication fork. On the leading strand, you'll have DNA polymerase 3 is going to follow helicase. And as it follows the helicase, it will add on the nucleotides according to the complementary base rule, like I mentioned before. So this is the easy part because it's going in the direction that it is comfortable going into. So this is your continuous replication. Now, on the lagging strand, however, DNA polymerase 3 will replicate short portions of DNA at a time. These are your Okazaki fragments, and this is your discontinuous replication. So the Okazaki fragments are going to be ligated together, which means to tie together, by an enzyme called DNA ligase. Okay, in order to terminate this process, because it's a single circular chromosome, what actually happens is you will get two circular chromosomes at the end of the replication process, but they're linked together, so they're interlocked. And we don't want them interlocked. We want them to be able to be separated so that they can become two separate cells in the process of their binary fission. So what do? So we must separate those out. So how does that work? So you have bacterial topoisomerase 4, which is going to go and induce a double strand break into each of the DNA molecules, and it will then pull them apart, and then that same bacterial topoisomerase 4 will then seal it up again. So yeah, so it's going to tear it apart, set it free, and then put it back together. And that's why this chromosome is saying freedom at last. Okay. All right. So that was DNA replication or DNA synthesis, which must be done every time we are going to have any type of cellular reproduction, because you don't want to have a cell without any DNA, or you're not going to get a cell because it can't function without it. So next, let's talk about the central dogma again of molecular biology and gene expression. So genes are segments of nucleic acids. These are sequences that code for proteins, and these proteins will end up exhibiting, some specific function in the body or some physical trait or something like that. The process by which the genetic sequences get expressed into protein is the two-step process, which I mentioned before, which is transcription and then translation. So in transcription involves using DNA molecules as a template to form messenger RNA. And the second and final step of the process is translation, which is where the mRNA sequence will be translated into individual amino acids, which will be linked together to make up a protein product. Okay, and like I mentioned before, here are those same three terms, okay? The three-step process for transcription is also initiation, elongation, and termination. So let's briefly define what those are. So with RNA transcription, RNA transcription in bacteria is done in the cytoplasm and not in the nucleus, like eukaryotic cells, because remember, bacteria do not have a nucleus. Transcription in bacteria is done using RNA polymerase. And the RNA polymerases, they are going to add nucleotides according to the complementary basis. And they add them on using the free hydroxide group that is just out there waiting for new nucleotides to be added. So how is this process of transcription initiated? So briefly, RNA transcription is going to require a promoter. What is a promoter? So a promoter is a DNA sequence, and the DNA sequence is required for transcription factors to bind in order for any transcription to take place. Promoters are going to be located what they call upstream from the portion of the amino acids, from the portion of the nucleic acid sequence that is going to code for the protein. The nucleotides that code for proteins would be considered downstream from the initiation site, which is where transcription will actually begin. All right, so taking a little bit more specifically, a little bit more specific look here at the promoters in bacteria. We actually have two different promoters in bacteria that are conserved sequences that we see throughout a lot of the different species of bacteria. So, and because they're conserved, we call these consensus sequences. So the two consensus sequences observed in bacteria are as follows. We've got the -10 consensus sequence called the Tata box. And we also have the -35 sequence, which is recognized and bound by the sigma unit of RNA. So let's go ahead and look at that. All right. So once all of the relevant transcription factors have bound to the promoter, elongation can begin. In this part of transcription, the sigma subunit of the RNA polymerase is now going to fall off. And when the sigma subunit falls off, this releases the RNA this allows RNA transcription to begin. The RNA nucleotides are added in a complementary fashion to the DNA template in the 5' to 3' direction at a rate of approximately 40 nucleotides per second. Now remember, RNA does not have thymine. That's one reason why I like this this figure here that I chose for you guys, because it shows the pairing here. So since RNA does not have thymine, when the DNA template contains a nucleotide with adenine, instead of getting paired with T like it normally would if it was in DNA, it will be paired with uracil instead. Here you see that abbreviated as U. Transcription of RNA then has to be terminated once everything is done. So once you have your RNA, the transcription will be terminated. And during this process, what occurs is the RNA polymerase will dissociate from the DNA template, and the newly made RNA will be released. This is accomplished through a terminating sequence, which is a repeated nucleotide sequence. And this sequence is going to signal to the RNA polymerase to terminate the process. All right. So once we have our RNA, the next process will be translation. So let's now look at how translation in bacteria cells is initiated. In bacteria, translation of mRNA into an amino acid sequence begins with the formation of what we call an initiation complex. The initiation complex is formed when the mRNA and tRNA and the first amino acid all come together within the ribosome itself. Once that occurs, elongation can begin. Elongation involves reading the nucleotide sequence of RNA. It reads it as a triplet called a codon, and you can see that here in this figure. All translation begins with what is called the start codon. So all of them will start with the AUG codon. So this means that the first three bases on the mRNA are adenine, uracil, and guanine, which is AUG. And the AUG codon is going to code for the amino acid methionine. Once the amino acid sequence is done, then we must terminate the process. This is also done with a sequence. This sequence is called the stop codon. There's actually 3 different codons that are considered stop codons, and these are the UAA, UAG, and UGA. So when one of these stop codons reaches the ribosome, the last amino acid will break free from the ribosome, and it will also break free from the tRNA or transfer RNA, and it will now be a free polypeptide or a free protein that can now go and do whatever it needs to do. Okay. Next, I want to talk about the genetics of viruses. Bacteriophages are a nice group to study. So bacteriophages are viruses, but these are viruses that specifically attack and infect bacteria cells. Viruses are not made of cells themselves. They can have either DNA or RNA as their genetic material. Since they're not made up of cells, they do not meet the current criteria for what we call life. So they're not considered alive, even though there is some debate right now that maybe we should redo that criteria because they're so highly complex. It's kind of hard to think that they're not alive because they're pretty amazing, which we'll look at here in just a moment. And also, they don't have the ability to reproduce without a host, so they are dependent on a host for their reproduction and all of their metabolic processes. Okay, so there are 2 different types of life cycles that there are 2 different life cycles that viruses and bacteriophages can go through. One is called the lytic cycle, and the other is called the lysogenic cycle. The lytic cycle is going to result in death of the host cell, whereas in the lysogenic cycle, the viral genome will actually be incorporated into the nucleus, and it will enter itself into the host genome, and it will keep the host alive for some time. In the lytic cycle, the lytic cycle ultimately results in lysis, or lysis means to cut, but you could also think of it as bursting open of the cell host, which of course leads in death. The virus is going to start the process by infecting the host, and it does this by usually attaching itself through a lock and key mechanism and inserting its genetic material in the form of either RNA or DNA into that host cell. At that point, the viral genome will hijack the host cell's machinery to reproduce itself and and it'll have the host cell make proteins and make the viral genome. And after everything has been made, the viral copies are then going to self-assemble and then leave the host cell, which results in lysis and death of the host cell. Now, in the lysogenic cycle, the viral genome is going to enter in the same way. It will be infected. But this time what happens is the viral genome will be incorporated into the bacterial genome of the host cell. And typically, this host cell will be kept alive for some time. The genome of the virus will be inserted into the genome of the host cell. So every time this infected bacteria reproduces itself, it's also going to reproduce the viral DNA as well, or RNA. After some time, eventually this doesn't go on forever, but eventually once it's kind of used up all the energy resources of that cell, then it will be called a prophage, and it will exit the host cell's genome, and then at that point it will undergo the lytic cycle, which leads to ultimate cell death. Okay. So bacteria cells reproduce using binary fission. So that means that when they reproduce, they are not having any sort of genetic recombination. However, we see that having genetic recombination and having genetic variances within a species is highly, highly important for survival of that species. So the way in which bacteria can have this done and do this naturally is pretty profound. So let's go ahead and look at this. There are 3 methods by which bacteria are known to undergo genetic recombination. This includes transformation, transduction, and conjugation. Transformation is not a natural process. This is a process that is done artificially in a lab. And what happens in transformation is the bacteria will take up a plasmid from the environment. In order to have this happen, the microbiologist must induce competency because they don't really readily want to take up plasmids that are found in their environment. There's a lot of different ways to do this. What we want to do is we want to perturb the membrane. We want to disrupt the membrane enough to allow the plasmid to get in, but not so much that we kill the cell. So it's a very fine line. There's a lot of different methods to do this. One of the most popular and easiest ways is to do what's called the heat shock method. So heat shocking is going to involve exposing the bacteria to a moderately high temperature for a very brief amount of time. This is under a minute. Immediately following the heat exposure, usually the culture is put directly on ice. And that extreme sudden temperature change is usually enough to perturb that membrane so that you can get a lot of uptake of that plasma into the cells that you want to have that plasmid. The bacteria are able to internalize these plasmids, and they can express the genes on those plasmids in the form of proteins. Okay. Another way in which genetic recombination can occur in bacteria is actually through viral infections. And this does happen in nature. It's not super common. It's a little bit rare, but it is a natural process that we've seen occur. So what happens here is phages or viruses can accidentally transfer genetic material from one bacterial host to the next bacterial host. And there are two different forms of transduction. We've got generalized transduction, which involves the lytic cycle, and then specialized transduction, which uses the lysogenic cycle. In generalized transduction, the phage is going to land on the bacterial cell. It's going to attach, usually using a lock and key mechanism. The viral genome will get inserted into the host cell. The bacteria, the host cell will then have all of its metabolic machinery and reproductive machinery used in order to make new viral copies. The new viruses or phages are then going to leave the hosts once they are all assembled. And what happens though is some of these phages might have some of the host bacteria's DNA inside of it when it exits. So if one of them happens to have some of the bacteria's DNA inside of it, when this phage now goes to infect the next host cell, instead of injecting its viral DNA, it will inject the host bacteria, its previous host bacteria genome instead. Okay. So in specialized transduction, this is similar. It's just using the lysogenic cycle. So here the prophage is going to leave the bacteria cell, and when it does so, it will actually have a small piece of the bacterial DNA in it, like shown here. And this bacteria will be transferred to any new host that the cell, that virus infects next. Okay. So next, let's talk about this happens very, very frequently in nature, and we see evidence of this happening all the time in laboratories. The process of bacterial conjugation. So in bacterial conjugation, this is not a form of reproduction, so keep that in mind. But it is a form of genetic recombination, which is very interesting. In bacterial conjugation, bacterial cells are able to exchange their genetic material. This is not done as part of their reproductive process. Conjugation is made possible using the F plasmid. The F plasmid is the fertility plasmid. It contains genes that allow the bacteria to form a sex pilus. Sometimes you'll hear this called pili or pili, which is the plural form of pilus or pilus. The pilus is a protrusion which can penetrate another cell and then penetrate the cell wall and the membrane of another bacterium, and it actually will create a cytoplasmic bridge between the two bacterial cells. Once this cytoplasmic bridge has formed, then the F plasmid will make a copy of itself, and one of those copies will be donated to the bacteria cell that did not have the F plasmid. All right, so that's all I have for you, and I think that was plenty. So good luck on your test.